pea plants grow? Well, you know, peas. What color are peas? Green. It's a trick question. Some of them are green, and some of them are yellow. Ha! It's true. And green is dominant. Now, has anybody here ever really spent a lot of time thinking or looking at peas? Thinking about or looking at? Probably not. I don't know why you people are wasting your lives. Just get out there and look at some peas. No. Peas also, as it turns out, and I'll draw both of these in green, peas not only have differences in color, they have differences in texture. And you have smooth peas, we'll say T for texture, little t for wrinkly. Smooth versus wrinkly. And they also, has anybody grown peas in a garden? Okay. I love fresh sugar snap peas. I could like eat my weight in sugar snap peas all summer. Clean them off the plant. Well, see, we don't get the kind you shell. I plant the ones that you can just eat whole, and they never make it in the house. They never get cooked. We just eat them off the plant. They're like popcorn. So anyway, peas have really pretty little flowers, and some of them are purple, and that's dominant. And some of them are white, and that's recessive. So since we usually name this for the recessive, we'll do that. So what Gregor Mendel did, because he was a little crazy and a little obsessive, is he crossed millions of peas. Millions and millions and millions of peas. You should probably be writing this down, guys. And so it is from his work that we started to understand, and I'm going to shrink little Gregor here, that if we take two pea plants that are both heterozygous for a monogenic trait like pea color, that out of that cross we get a well, he didn't know about genotype ratios. You know why? Because he didn't know about genes and he didn't know about DNA. He did all this without understanding that there was DNA or chromosomes or how any of it worked, just based on stats. He was a little obsessive. Some of the best scientists are. But what he was able to understand was that he said that there must be factors. He called them factors because he didn't know what genes or anything was. And he noticed that if you compared green to yellow, you got 3-to-1. You knew that, right? So we've done monohybrids. You get 3-to-1. So roughly what percentage of the offspring show the dominant characteristic? About 75%. 3 out of 4. Okay, well, that's easy. So if you look at any monogenic trait all by itself, for some species, you'll see that roughly, Gesundheit, roughly 75% of the population is going to show that dominant trait. But what we want to think about next is what happens when we look at the two traits together. And this is what we started yesterday with the dihybrid cross. So we're going to forget about the green and the yellow peas. Well, we're not going to completely forget them. But we're going to look at what happens if we look at that along with another characteristic. So those are the three characteristics or traits that we have to examine. Let's do flower color and pea color. Okay. So if we have two plants that have purple flowers and green peas. And they are both, what's that term when they have two different alleles? Heterozygous. I guess I could, well, that's okay. I can do this in black too. They are both heterozygous. Oops, sorry. Now, when plants make gametes, it's pretty much the same process that we looked at in mammals. Now, there it's eggs and sperm. Here it's eggs and pollen granules. So we don't get to draw fun little tails on them, but that's okay. Every gamete that that pea plant makes, whether it's egg or pollen, just thinking about this one, it's got a 50-50 chance of getting which one? 50-50 chance of getting a big pea, a dominant. It's got a 50-50 chance of getting a little pea, recessive. Each gamete that has a big P has a 50-50 chance of getting a big G. Each one has a 50-50 chance of getting a little g. For the ones that received this small p, 50% chance big G, 50% chance little g. How many possible gamete combinations do we have? Well, 1, 2, 3, 4. This is going to give us one of those monster 4x4 dihybrid Punnett squares. So, we're going to put each possible gamete combination across the top. Big P, big G, big P, little g, Gesundheit. Little p, big G, little p, little g. And we're going to copy those same exact things right down the side. So I've got my giant chart here. Oops. Move it kind of front and center. Now, do you feel pretty confident in pulling down those alleles? I'm going to do it slowly. You can watch what I'm doing, but when you think you've got it, you just fly on your own and pull down the alleles. I'm going to pause this. Resume. I want to record that. So yeah, this is 2 traits at a time. That's why it's called dihybrid. And we separated. These all show what's in the gamete, which is only half of the genome. When you put them together, you get individuals who would have a full set of alleles. They would have 2 alleles.
See, I thought this was just really polygenic traits.
No. Oh, God, no. That's so far above our pay grade, we can't do that. And it's an interesting question. Oh, that's a really good question. So, note that you are putting the alleles back together the way that we normally show them in an individual once we get down into the boxes. Okay, so if we want to know how many, if I want to know how many of these have purple flowers and green peas and purple flowers and yellow peas and white flowers and green peas and white flowers and yellow peas, those are my phenotypes. Most commonly, I have to list all my genotypes first. Because the only way to really figure out, well, how many of this phenotype do I have, is to go through that genotype ratio. Does anybody remember from yesterday what these ratios look like? There are 9 possible combinations. So big P, you know, big, 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 big. I got 1. Big, 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 little. 1, 2. Big, big, little, little. 1. Hey, look! That's a square! Huh, that's weird. Okay, big, little, big, big. Big, little, big, big. Big, little, big, big. I got 2. Big, little, big, little. 1, 2, 3, 4. Big, little, little, little. 1, 2. If you were a betting person, would you care to bet what the ratios in that bottom one are?
1, 2, 1.
1, 2, 1. Yeah. And we can prove that. We can go, okay, there's little, little, big, big, 1. Little, little, big, little, 2. Little, 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 1. But what we really want to know is what number have purple flowers and green peas. Well, we know that everybody up here is going to have purple flowers, right? Because they have at least one of the dominant alleles. Yes? Okay. And we know that these guys and these guys and these guys and these guys all have green peas, yes? So that's 3, 6. So we have 9 individuals with purple flowers and green peas. And we know that these guys and these guys have yellow peas. So that's 3 individuals who have purple flowers and yellow peas. And then everybody down here has the white flowers. And these 3 have green peas. And there's just this one poor little pea plant that's got a white flower and a yellow pea. The one kid in the family everybody else picks on. It's just different than everybody else. It's the tiny little percentage. Well, do you think Mendel bred 16 pea plants? No, he bred millions of pea plants. Millions and millions and millions and millions. So, grab a calculator. There's one on that desk. They're right behind Hannah Brennan. Here, pass them around. Take one down, pass them around. 53 calculators in this room. We would expect that 9 out of 16 would be dominant for both characteristics. Huh. Roughly 56%. 3 out of 16 would be dominant for flower color, recessive. It usually rounds to about 19. It's 18.75. So about 19%. Well, we know this one's going to be the same. What about the white flower yellow pea? That poor little individual there. About 6%. Yeah, everybody picks on them. So here's the thing. When we look at 2 characteristics together, we can assume that if those are monogenic characteristics, about 56, if we look at those traits together, about 56% of the population is going to have the dominant version of both traits at the same time. So, 75% of the population will have at least one of the dominant traits. But if we look at those traits taken together, we're seeing that over half the population will tend to have both dominant characteristics. So let's look at an actual sort of genetics problem. Who here has ever seen a guinea pig? They eat them in South America, right? They're livestock. They're a little bit bigger. They're about 5 pounds, but yeah, they're just another farm animal in South America, in the Andes. Sorry if you didn't know that. Let's talk about guinea pigs. Okay, so these are guinea pigs, and guinea pigs have 2 options for fur color. They can have brown fur, or they can have red fur. And they have 2 alleles for fur texture. Curly or straight. And this is true, and if you've ever seen a curly-haired guinea pig, they are hilarious. They just look, I don't know, they just look like they were put to bed wet. They're just all like crinkly and it sticks up, and they're like an entire body little rodent covered in cowlicks. I find them hilarious. So, let's say that some crazy geneticist somewhere breeds 1,000 guinea pigs in their crazy guinea pig barn. And let's see. "57 of them have straight red fur. And 211 of them have straight brown fur. And 552 of them have curly brown fur, and 198 of them have curly red fur. So we have roughly 1,000. I don't actually know if my numbers add up to 1,000, but they bred about 1,000 guinea pigs. So you guys think that brown is dominant. What about texture? Curly? So 211 divided by 1018. Huh. Well, that's 21% of the population. 198 divided by 1018. That's 19% of the population. 57 divided by 1018. Oh, that's 5.5% of the population. Huh. Does that start to look like the percentages that we saw here? Where we expect that if we have two dominant traits that are being inherited independently, about a little over half of the population is going to have both dominant traits. Now, this assumes something. So yeah, we can look at a population breakdown for two traits, and we can pretty well guess which ones are dominant. What this assumes, so you all remember when we made chromosomes, and I'm going to go with all sorts of wonderful sexist gender stereotypes. For each chromosome an organism gets, for each chromosome you got one from yo mama and one from yo papa. Because I have fun saying yo mama and yo papa. I don't know why. So you've got a maternal and paternal copy of that chromosome. And they've got all the same genes on it. And here, we'll put a little gene right there. So if we're looking at one gene all by itself, you know, it gets sorted, it gets mixed up. Now, do you remember when they did that weird thing they did? That really weird thing they did where they copied, they copied themselves, and then they hooked together in the middle, and then it got even weirder because what did they do next? What did they do next? They wrapped their arms and legs around one another. You remember that? And they tore off some chunks. So then suddenly we end up with the dad chromosome having a chunk of the mom chromosome on it, and the mom chromosome having a chunk of the dad chromosome on it. Well, now, if you had a gene that was here on each chromosome, and you had a gene that was up here, those genes wouldn't necessarily be traveling together, would they? They'd get split up a whole lot. This is that crossing over and they recombine. You get new combinations of genetic material. What if, has anybody here ever gone to a concert or a party with a friend and lost them? In the old days, before we had cell phones, and you couldn't call your friend and go, no, I'm standing under the sign that says 4E. I can see you. I'm over here. You couldn't do that. There were no cell phones. You just spent the whole time walking around looking for your friends. Oh my God, where are they? Okay, if we get lost, meet at the snack bar. And you just had to meet somewhere. Well, if you've got a gene up here and a gene here, is it likely that they will get separated from one another? Yes. What if you have a gene that's right next to another gene? Is it less likely that those two will get separated? Yes. So in these cases, we're looking at genes that are what we call independently assorted. They're not necessarily traveling closely with another gene. So they randomly assort, and you can get the dominant or the recessive, and there's no rhyme or reason. But sometimes we have genes that are close together. We call this a gene linkage. And actually, we've been doing Punnett squares. Did you know there's actually a scientist named Punnett? Reginald Punnett. Guess what? He was the first person to use these funny little statistical boxes to figure out probabilities on genes. So Punnett and, Saunders and Bateson. Punnett, Saunders, and Bateson. At one point, they were studying sweet peas. I'm going to shrink this. And they were looking at sweet peas which have, red or purple blossoms, and the purple is dominant. And they have either long pollen capsules or round pollen capsules, long as dominant. And they crossed a bunch of these. And they expected to see that, 56%, 19%, 19%, 6%. Here's what they got. So for purple, long, and then you have, of course, let's see, purple round, red long, red round. So what they got instead was 284, 21, 21, 55. So let's do the percentages real quick. 284 plus 42 plus 55, 381. 284 divided by 381. Well, that's weird. 75%. What? That's supposed to be 56%. And 55 divided by 381. This is 14%. 21 divided by 381. This is 5.5%. That's weird. Do you know that the moment right after somebody says, oh, that's weird, is when the best science happens? Because when you get something you're not expecting, either you've really screwed up and you should rerun your results, which you should rerun your results when you get something really weird. But very often when you get something really weird, what it means actually is that you're into territory that nobody was aware of before. So it turns out what they had discovered was what's called a gene linkage. And, the genes for color, flower color and pollen shape were linked. And the dominant traits for that almost always went together. So you see a much higher percentage than you would expect of dominant, and a much higher percentage than you would expect also of recessive. Because we would expect to see like 6% recessive. We're seeing 14%. What they discovered in this was that some genes are linked, and they now, as a standard course to prove gene linkages, well, prior to having the whole human genome mapped out, they would actually run multiple crosses and look at the percentages. And based on how far those deviated from the expected percentages, you can figure out how close on a chromosome those two genes are, which is pretty amazing. Now, two genes that are on completely different chromosomes are never linked. Two genes that are on opposite ends of the same chromosome are very rarely linked, probably never. Genes that are close together on a chromosome can show linkage, and you can figure out how close they are based on how linked they appear to be. Okay, here's the deal. You're going to have homework. So we are down to crunch. We are down to, what, 5 instructional days, and they're blowing one of these up for some speaker because that's really critical. I'm really excited. You can tell. I'm sorry. You're going to have homework probably every night between now and then. I have been super generous all year. You have seldom had homework in this class. You got about a week of homework every night. Suck it up, Buttercup. And then once we're through the tests, we'll not have a whole lot of homework after the test. We're going to do some experimental stuff and some kind of fun units, but you are going to have some homework between now and then. So it is going to be posted on Classroom. It is an electronic assignment. If you have a stay-at-school iPad, talk to me, do it in a study hall, work it out, or I can print off paper copies. But right now, these are just 2 air test questions that relate to this. You can probably bang them out right now, or maybe in your 7th period class. So that's it, folks.